Maybe people got confused. We know like in Houston, trying to move their offices. We almost got confused, but it's not now. That's really well. My rule of thumb is one person. This is like spring break. Or you still have
Spring break is over.
oh, maybe soon this week, right?
A couple of weeks ago, I think.
Oh,
Yeah.
like the UW is this first week.
Oh, I see. I see. Cool. Yeah.
I think because of how fast you guys kids will see, I don't what is about snow, we will have a lockdown pretty much.
For sure. Yeah, they freak out because they've never drove in that condition before. Here, we're more prepared. We have the infrastructure to handle snow.
Not because I always get surprised, right? So much Utah, we get snow every year. Right? So like in my head, I'm like, yeah, we know how to drive in snow, but I swear it's like half the drivers on the road, like you can tell, yep, they are driving this, and then there's like the other half, and I'm like, how long have you been living in Utah? How do you not know how to drive in snow yet? But yeah, it's good people, especially when it's like this late. Wait a year, it's not late in the actual year, but like wait in the season. We're like, oh my gosh, they're supposed to know.
Yeah. Use winter change my winter tires back to
Yeah, seriously, I don't even have winter tires, I probably should, but yeah, but I always drive my all-wheel drive automatic when it gets like really bad, because my other cars are not all-wheel drive and they're manual, so with the hills up here and the snow. No.
How are we doing with the audience?
Yeah.
Getting there.
That's good, some of the next students too, just there for my own. So on my audience, so it's obviously all the Center for Global Surgery, everybody in molecular pharmacy. I think we blasted it out to our MSCI student. 3 or 4 other different members.
All right, I think we should probably just get started because there are a bunch of folks online. But I know that a lot of our folks are all around the country and world sometimes. And it's just a matter of who's physically able to get into the university today.
Cool.
Was all this.
Can everyone hear me on Zoom?
Yes.
Awesome. Hello, everyone. I'm honored to introduce Dr. Nuttada Panpradist. Dr. Panpradist is a postdoctoral fellow at the University of Washington in the Department of Global Health. She's also a research officer at the Kenyan Ministry of Health and a faculty lecturer at Chiang Mai University in Thailand. She obtained her PhD in UW in the Department of Bioengineering, and she is committed to making healthcare accessible and affordable. Her research is in the development of innovative molecular diagnostics for applications in global health, and she has developed rapid tests for HIV, tuberculosis, drug resistance, and COVID-19. She has won countless number of awards and grants that recognizes her research and leadership. A few recent ones include 4 new investigator awards from the UW WACh Center for AIDS Research, Seattle TV Research Advancement Center, Thrasher Research Fund, and the UW Institute of Translational Health Sciences. Please join me in welcoming Dr. Panpradist to share her work with us today.
Well, thank you very much, Dr. Ying, for the kind introduction. Hello, everyone. It's really a pleasure to be here. Once again, my name is Nuttada Panpradist. I often go by Dr. Kanda. And I think through the introduction, you might have heard already that I work with so many people and so many disciplines. And I think that's a part that made me decide to join the Global Health Department right now, because it actually conducive to the collaborative environment. And not just working across discipline. One thing that I realized that is considered very important to work with people across the globe is to engage yourself, embrace yourself in local settings. And that's why the position you see in front of you on the slide is actually the position that I accept in Thai, so that I can actually serve as the co-advisor for these students in different universities. So you see here for the Associated Medical Science at Chiang Mai University in Thailand, Forest Biology in Kasetsart University, and also serve as the research officer in Ministry of Health in Kenya. And I always give credit to, the reason why I'm able to collaborate with people from across background and also from different disciplines is thanks to my early exposure to, let me see, so multidisciplinary education and training. So I received my first training in engineering background in Thailand. Then I realized that my goal is to try to make healthcare accessible for all. Unfortunately, at that time, Thailand didn't even have bioengineering field, so I came all the way to the US to pursue this goal. But jumping the field is not easy, and also when you change the culture, it's also not very easy. So I have to get myself prepared and take some courses to learn more about healthcare system and also learn how medical devices are actually fabricated through some certificate degree. Then later on, I started my PhD in the Department of Bioengineering under Dr. Lutz, who worked on expanding access to healthcare, especially in the field of diagnostics. But in the middle of my PhD, I also, it's more like I'm a long life learner. I realized what is missing and I just continue to educate myself. So I received a six-month fellowship to allow me to explore new area of research, to visit Stanford University working with Shirit Inab and Stephen Quake to learn about single-cell RNA-seq so that I could learn what are the biomarkers that are behind the information of dengue infection over time. And also received the Global Health certificate as part of the adolescent children and women. So that's a lot of background, but that also explains the following slides just to summarize some of the work that I did during my PhD and also 15-month postdoc. I have done quite a bit of work, and today I won't be able to tell you all of them. So I selected one project that I spent a lot of my time working on. It's called Simple, which is a single-base mutation detection platform. And I just want to highlight maybe on the left side below, those are funded research projects that I initiated. So I came up with the idea during my PhD and, wrote majority of the grant, and later on it turned into a funded project. And then the one highlighted in blue, those are the ones that I wrote indefinitely as a postdoc. So it's quite very fortunate that I'm able to come up with a good idea and pursue the funding that can support my work. And one thing that I could not, stress enough is about the importance of undergraduate researcher. For me to be able to work on these many projects and keep things going in a way that is smooth is that I have been, it's a pleasure to work with a very talented, dedicated group of undergraduate students. So during my PhD, I worked with 9 students, and now as postdoc, I work with students from different departments, and there are a total of 15 students. So enough about introduction. I'm going to dive deep into the technology I'm telling you today. Simple. It is the platform that can detect single-base mutation. And the reason why I picked this project to share with you today is because I saw that global health people not just care about technology development, but how it can be translated to potentially make an impact in the field, right? So as a student, I spent most of my time on this project, and it was amazing to have experience to be exposed to working in the environment that start from ideation, development, scale up your techniques, and allow for validation demonstration in the field before even doing publication. So I think I'm gonna just dive in on what OLA symbol is. OLA symbol is a technique that can detect single base mutation. So single base mutation, sometimes referred to as single nucleotide polymorphisms or SNP, is one of the most common form of genetic variations. And here you can see example of one where AT changed to gene C right here. Access to SNP information can be really useful as SNP has been proven to be associated with many genetic diseases and conditions. In particular, SNP info can guide clinician to personalized treatment. For example, you see here, this antibody treatment binds to SARS-CoV spike protein and prevent the virus from entering into the cell. However, if there is a SNP right here, it will escape the antibody recognition and enter into the cell. Thus, personalized treatment based on SNP info can improve treatment efficiency. Another example is a person with this particular mutation would have hypersensitivity to the drug named abacavir. Thus, personalized treatment based on SNP information in human genome can also ensure patient safety. Unfortunately, obtaining SNP information could take a very long time depending on where you are in the world. And this number right here is based on when I conducted a survey from different countries around the globe. It takes this long depending on where you are in the part of the country as well, because sample need to be shipped to the clinic or hospital and from the clinic to the centralized lab for processing. And this long wait time could lead to patient stress, loss of follow-up cases, improper medication use, and if this were to be infectious diseases, it could lead to public health risk. So our group had a vision to develop a decentralized test that can detect the key SNPs and allow for clinicians to take actionable information to personalize treatment at the point of care. And my PhD thesis focused on improving speed, simplicity, and affordability of an existing SNP detection workflow. Our first application was focused on HIV, which has been one of the biggest pandemics before COVID-19. And even though HIV, as you may know, that it has been around for nearly 4 decades, we're still fighting with it, right? And part of this prolonged pandemic, especially in low to middle income country, is because of drug resistance. Nearly 2 years ago, you can see the data here that 1 in 4 adults and 3 in 4 infants that are born to seropositive mothers, even before they start their treatment, they already had HIV that is resistant to the first line in an RTI regimen. Therefore, to ensure effective treatment and prevent further transmission, HIV drug resistant test or HIV-DR test result can be used to personalize treatment therapy. The test for HIV-DR we can actually look at the key SNP information that are associated with treatment failure. So these SNP that are actually introduced by the RT enzyme of HIV during the replication process. If some of these SNP happen to be at one of the hotspots, like you see here, where A at the position 181 change to G, it will alter amino acid sequence that cause mutant HIV to become resistant to this drug nevirapine. Okay. And it has been shown that 6 SNPs are associated with HIV-DR against first line in an RTI. To look at this SNP info, we can actually look in either RNA or DNA because HIV is a retrovirus that targets CD4 positive cell. So after the membrane fusion, the RNA is released and converted into the cDNA, which later integrated itself into a human genome as a proviral DNA. For pre-treatment population, RNA and DNA contain similar information, and DNA is actually less prone to degradation. So in terms of assay development, we can target DNA. However, if you are going after the population that already started treatment, RNA would be a better representation for biomarkers for HIV-DR testing because that actually picking up the virus that are actively expressed. Therefore, an HIV test for treatment optimization for that geared toward LMIC should be specifically discriminate at least 6 key HIV-DR SNP that I told you about. It also, if you look closely, there are the blue base right here. This represent nearby mutation, or sometimes referred to as like nearby polymorphism. So when you design an assay, it needs to be able to tolerate nearby polymorphism as well. HIV-DR test should also detect down to 10 to 15% minority variant in a single person. This is very different from the case of tuberculosis or COVID, because one person is usually infected by one variant, whereas HIV is what we call quasi-species, so you get multiple types. And to be able to look for the HIV-DR that are linked to drug resistance, you have to be able to detect down to the 10 to 15%. And this is exactly where Sanger sequencing method falls short, as it cannot detect low-level minority variants. And on the other hand, oligonucleotide ligation assay, or OLA, can actually have great analytical performance for both sensitivity and specificity, as you can see here. So what is OLA? OLA high sensitivity rely on the polymerization reaction, or PCR, which you all probably have heard about. During this process, the region that contain HIV-D are amplified from a few hundred copies to billion copies. Then this is where it's unique for OLA. The high specificity of OLA relies on ligase detection reaction, or LDR. Here we have two probes, the left side and the right side, that targets each side of the HIV-DR SNP. Each has a different N label that you can see here, FAM and biotin. And only when the bases are matched at the target SNP, the ligation will occur where enzyme ligase will covalently link the two probes. And ligation of the probe will not occur when the bases are mismatched right here. So only ligated probe can then that has two N labels can then be detected using the standard enzyme linked amino assay, or ELISA. One N label would be captured onto the plate, because you can see here, and on the other end would be recognized by antibody enzyme conjugate, which can create a signal in the presence of specific substrate. Our collaborator, Dr. Franco, who is a really renowned, well-known HIV clinician who worked in this field for more than 4 decades, have pioneered a version of OLA for HIV-DR. And the key change that she made is this version also includes a probe that target the wild type, not just mutant version of HIV. This is a very important addition because it will check if ligation failed due to the new polymorphism that in the specimen with 0% mutant. Additionally, LDR also operate at much lower temperature in order to allow the probe to tolerate nearby polymorphism. So now you have two types. You have both mutant and wild type probe that are ligated and are captured and detected sequentially by ELISA for mutant and wild type signal. So there's a lot of chemistry here, but just big picture for you all to catch up is that it's really quite a complex, even though it's pretty sensitive and specific. The complexity itself actually limit the wide adoption in low resource setting. There was several attempts to roll this out in Kenya and China. It's only limited in really high facilitated lab, but not beyond that. And the process itself takes a really long time, like 9 hours from sample to result, need 10 personnel and also equipment such as plate reader, plate washer, and also a thermostator. So to expand the access of OLA, the Franco Lab collaborated with the UW Bioengineering to develop OLA sample. And over the year, we were able to grow our collaboration that cover different areas of the work that you see here, from assay development, interactive software guidance, industrial collaboration, and also representation of clinicians from around the globe, because HIV is one of the most highly mutation, right? So you want to make sure that your research is inclusive and have representation from different subtypes. So I'm going to tell you first version of what we call minimal viable product, or MVP, which is a simplified HIV-DR test for small hospital lab that target the P30 HIV-DR that can detect across subtypes. So there are 3 components for this. It's a simple sample prep, ready-to-use dry reagent, and also lateral flow device that allow for simultaneous detection of wild type and mutant HIV. And first, we'll tell you about the simple sample prep. So if you recall a slide a while back, only CD4 positive cell would be infected by HIV, right? So only this cell would contain HIV DNA. The problem is that when you use traditional sample prep method that pull out total DNA, it have really low mass to ratio of HIV DNA to human DNA. So the previous version of OLA was able to overcome this issue by doing 2-round nested PCR to improve sensitivity. But that takes a really long time. So one trick that we use to improve the speed is to use CD4 negative cell depletion. So we use magnetic bead that have coated with antibody cocktail that designed to bind everything but CD4 positive cell. So this complex can then be removed by magnet and allow us to have the HIV DNA rich portion for downstream processing. And we were able to pair this with the in-house low-cost magnetic rack and generate a workflow that enables sensitive and single-round PCR. So that's the first part of sample prep in the first version of our kit. Then the second part of the component of the assay is ready-to-use dry reagents. Setting up a molecular reaction requires precise measurements of multiple reagents. For those who set up PCR before, that's you can see, but I think for ligation, this is one of the actual components. You can see how many it is. To overcome the complexity, we collect all these components into a single dry reagent pellet that our user need to do is just add water and add sample. And also because of it become dry lyophilized format, it can be shipped without cold chain requirement as well. That makes it a lot easier to handle, distribution of the test outside the US. However, removing water from enzyme passively can really lead to an enzyme malfunction because naturally enzyme is surrounded by water molecule. And therefore, to maintain enzyme function, we have to use some trick, like for example, excipient formulations that contain saccharide to substitute more water molecule. To avoid structural collapse, we use bulky polymer chain, and we also use polymer with high glass transition temperature to promote long-term stability. We also use lyophilization process to prepare the dry reagent. In order to minimize molecular mobility, the mixture is quickly frozen in liquid nitrogen, and then ice is removed via sublimation. To avoid melting during the drying process, the transition from step 1 and step 2 that you see here, from frozen to drying, need to happen at temperature and pressure below the eutectic point, so then it transition from solid to gas right away. And I think this is a cute slide to share. We also develop multiple dry reagent platform. The reagent that can be prepared in tips that allow for any reagent to just, you can imagine if you use pipette tip, you can take this tip and then scrape the wax out and then just mix simply by pipette. Or if the excipient formulation is actually intact enough, we could prepare the beads, and these beads can be like pick and place into different devices. And the last is lateral flow strip. So this is many slides ago, but just to remind you, this is the last step of when ELISA happened, we have to have 2 probe capture onto the plate, then being detected by different antibody conjugate followed by substrate, right? But to make this step simpler, we first inverted the stack and have wild type and mutant product capture on different locations using different antibody, then add the control line. And then we can actually use the common label antibiochemical nanoparticle to label all of them, mutant, wild type, and control. But the main challenge here is that the false signal from unligated probe that can hybridize to mismatch template at the end of the ligation step can actually give you false signal. Like you can see here, a ligated probe from hybridization with the mismatch template. So ligation didn't happen, but it still can give you wrong signal. So we have to actually destroy this duplex. And the way to do this is to adapt the new probe design in combination with topo mediated stem displacements. So what you can see here is that we have engineer adapter that look like blue right there. And then we throw in competing oligo that would preferably bind to our unligated probe and ligated probe. And now it kicked off from the original template. So now you can see that competing oligo in yellow allow for us to differentiate between ligated and unligated probe. So at the end, you could see the result that could look like this, that have mutant mixture with wild type, And since some rare patient, is it very important for us to know if our probe are not working properly. You will see indeterminate result where ligation actually failed, but you will have the control line. So that is an indication that it's time for us to revisit the probe design. The virus have evolved too far, and we have to. This is very important when you kind of launch the assay in the field. So here's one example of the result. So, after those three components we put together is MVP1, OLA simple for DNA kit that have the following component. Is to rely on a thermocycle, its points, and it takes quite long. As an engineer, 4 hours is really long time, but it was 50% discount from the original 9 hours. But one thing that's really important, and I did this, actually took it all the way to present at the international stage at the 2017 WHO meeting in South Africa. And that really changed how I define my PhD. Because to my surprise, clinicians were not concerned about that 4-hour wait time, because they said that typically patient already have to wait 6 hours at the clinic. So I can find a way to actually engage with them, and 4 hours is enough to actually get the result in one day. However, we receive additional copious feedback that actually help us frame our research questions. And here are some research questions. So question number 1 is whether the current kit can detect that result in multiple subtypes of HIV, right? That which lead to the clinical validation across subtypes. The second question is whether the amount of training required for, how much of the training required for first-time user to do it, because you can see there are quite a bit of losing piece. So this leads to the development of software companion and follow-up usability studies. And the last question is it like almost a multi-million-dollar question. Where can I buy this test kit? When you develop the test in the lab, how the heck you can get it to the actual user and start making an impact? So instead, we start to answer many questions like maybe start to engage with industrial manufacturer and think about potential users. So that's our first step, to try to make prototypes enough that we can actually conduct the initial studies. So of course, we fall on to our first option. Let's team up with the undergraduate student, and everyone was happy to be a part of this. They made this kit, we made it all in the lab, everything in the lab to allow us to do some usability testing. But later on, we took advantage of a really great network in Seattle. We actually was able to identify 2 industrial collaborators that successfully allow us to transfer the protocol in a way that can scale up production very largely. And then these are identified by me and a group of students who participate in business competitions. So now that we have, a lot of kits that we can conduct a study, we went on and answered our first question. Can OLA simple perform well on archive cross-subtype HIV specimen? So this is thanks to a multi-continent collaboration. We were able to create a panel of samples with multiple HIV subtypes and blindly tested these samples using OLA simple. So lateral 4-strip test result were classified blindly by the untrained operators. And then we took the call from 10, 11, 13 people and call that a mode call and compare that result to Sanger sequencing or other sensitive comparators such as original OLA and MySeq, which can be taken down to 2% of minority variant. So very low level of minority variant. The answer is yes, OLA simple has a high performance in cross-subtype clinical validation. It received almost 100% sensitivity, specificity. And when compared to Sanger sequencing, OLA detected the mutant sample that were missed by Sanger sequencing because that's what we expected. And when we also found the level of illuminate at 2.6, which is very similar to what OLA simple would have. So that's actually quite nice. However, when actually benchmark it against sensitive comparators such as OLA and MySeq that can go down to 2%, we found that OLA simple has a sensitivity of 86.2%. So, we set out a question and try to understand where did the sensitivity drop. So here, when we actually do the plot, we investigate very closely to understand more about the mutant sample that were missed by OLA simple, indicated as the pink circle here. If we look at the x-axis here, the copy of HIV template per reaction did not seem to impact the ability of OLA simple to detect mutant sample. However, if we look at the y-axis, across sample subtypes, OLA simple missed more sample below 10%, right? and which is clinical relevant concentration. And when applied the same threshold for ampicon, it also the same. So if you actually do the adjustment of 10% cutoff, OLA sample has a 99.4%, which is adequate for clinical HIV represent test, whereas sample sequencing only have 89%. So that's good news. Next, we realized that the use of mode calls by asking 11 to 13 people to, analyze the result, it's not practical in real world. So we designed a survey to just understand how well non-trained operator can actually classify the result when being randomly presented with these strip images. We found that only 88% of strip were classified correctly by 90 to 100% of people. So to address these user variations, we have developed the software to classify the OLA simple results, and it is used as part of an Aquarium interactive guidance software. So we collaborate with the Clavin platform, electrical engineering and computer science to develop the interactive software guides for OLA simple that allow me to track sample, and the user can follow the protocol without extensive training, and also the result will automatically analyze. So based on the use of Aquarium and OLA simple together, we were able to recruit 41 first-time users. Some of them don't even have pipette training before, and those of them will go through Nuttada training for like 5 minutes. Okay, let's practice how to pipette, and then I walk away. And these people were assigned 2 blinded samples to them. And so they have to test the total of 16 mutations, and at the end make calls, both visual call and software call. It turns out that first-time user accurately genotype DNA at 97% accuracy. And at the time, we were really excited that, this is amazing that even though the lab test still have some losing piece, but when it's paired correctly with the software, you can actually enable first-time user. And then we didn't stop there. We actually took it all the way to do something similar at the clinic in Kenya. And for Kenya, we actually included a sample PEP step as well. And we were surprised and happy to see that we were able to achieve the same level of accuracy at 97% even in Kenya. So we went back to the WHO meeting the following year and say, yes, OLA simple work across HIV subtype. Yes, OLA simple successfully can be performed by first-time user. And for the last question, we actually reframe it to identify where OLA simple would be most useful. And we have identified the first location for OLA simple to be useful, which is Mexico. So in 2018, I believe, Mexico was one of the countries with high prevalence of P-therapy HIV-DR, but was not able to switch to the non-NNRTI regimen right away. So they would likely benefit from our technology. Importantly, Mexico is also has one of the WHO certified laboratory for HIV-DR testing. What it means is that it would allow us to compare the performance of OLA simple directly to the top-notch World Health Organization certified results, right? And this is all free by collaborating with them. So after the initial phone call and a follow-up meeting in Mexico, and a little bit of charcoal here, we were able to follow collaboration with the CNA in the WHO certified lab in Mexico. And we had to do some modification of the test to make it work for RNA sample, which is the sample that I used in this center. I'm not going to go into the detail, but happy to answer the question of what modification we did. But I think big picture scope here is that our MVP to OLA simple kit that are appropriate for RNA actually compare very favorably to MySeq that performed by the WHO certified lab in Mexico. So if you take the 2% threshold, you see 93.2% concordance, but actually at the clinical relevant 10% threshold, we will compare pretty nicely still at 97.4%. So just to summarize everything, these are we were able to significantly improve the ease of use and eliminated the need for plate washer and plate reader, and a thermocycle, which is still now the only remaining equipment that has recently become pretty inexpensively this day, and the protocol is 50% faster. And you might be asking where it's going right now. As we speak, we just finished the clinical study where the lab at 2 national lab in Kenya processing this sample. And now we're launching the other 2 studies in Nigeria and South Africa. And the clinical lab continue to identify key mutation and design the new probe that associated with other drug classes for HIV. The next question is, can OLA simple be useful not just for therapy treatment, but can be used full for monitoring treatment? So HIV testing can be useful for people who fail treatment as well. However, regardless of how much we could further simplify the OLA simple, its use would be limited by the access of viral load testing, as you see here. This is because patients need to be confirmed for viral load above 1,000 copies per mil before proceeding for HIV testing. And these 2 are currently done separately, which altogether would lengthen the patient wait time to access to their personalized treatment. And that's why I'm going to introduce you the 3rd MVP, which is BOLA. Is it an integrated workflow for viral load test and a reflex HIV resistant test? So we envision a workflow that can involve a single sample collection, and then which first will be tested for viral load. And then if the viral load above 1,000, the same reaction tool that you see there would go directly to the downstream process for drug resistant testing. So you can find out whether a person who have failed their treatment is because they are not taking the drug or because they have drug resistance. So that is the question that doctors always have. So to do this, we have to ensure that assay provide fast turnaround time to meet requirement of viral load test. And the viral load test itself need to be quantitative. And the back end of the HIV test need to be chemically compatible with the viral load test. And most importantly, in order to prevent contamination from PCR, you might have heard this before that when you run PCR, it's easy to do the first time, but to keep the performance across months, that require a lot of maintaining the no contamination from the previous run of PCR. So how do we do all of this without opening the tube? So to address the slow turnaround time, we first investigated and found that thermal cycle using PCR and the LDR step is a slow process. During the LDR step, all the components won't react until the temperature is raised to denature the DNA, and then cool down to allow the ligation to occur, right? And this cycle happened 10 times to generate detectable level of ligated product. It turns out that up to 60% of assay time was wasted on the heating and cooling down of the heat block. So our approach for this is to use isothermal reaction which occur at fixed temperature, so no heat up and cool down, just fixed temperature. And for viral load test, we rely on thermophilic helicase treatment application, or THTA, which contain 2 enzymes here, helicase and polymerase. We use a symmetric ratio of primer with a higher reverse primer concentration. We also use the dye to track the presence of the amplified targets. First, helicase would unwind the DNA and allow for primer to anneal and get extended by polymerase enzymes. Dye then bind to the amplified target and emit the signal. Helicase is doing its job in the second round, but now the forward primer is used up, so only reverse primer is available to anneal to the template and get extended. So at the end, we would have traceable amplification for quality viral load test above there. And also open reverse standard is readily for isothermal LDR. If we toss the LDR probe here, the probe will readily bind to the template without the need for thermal cycling. So next, we have to ensure no tube opening after the viral load assay. So what happened? We just combine amplification and ligation reagent in one tube, right? That's a simple question. Turns out the problem is that ligation probe can then be used by polymerase as a primer, like you see here. So at the end, we have failed ligation. So to overcome this, we have investigated and found that 1, THDA reaction use salt, low salt, while LDA use high salt concentration. So during the LDR reaction, THDA polymerase will remain inactive and cannot extend our LDR probe. Next, we also use the probe that have much lower melting temperature than the THDA reaction temperature. So the LDR probe will stay unbound and also won't get extended during the THDA polymerase. So here is the way we do it. We have one lyophilized reagent that contains isothermal reaction for viral test, and then ligase enzyme, LDR probe, and the dye, except the salt is dry on the lid. So after 50 minutes, we could see the amplification down to 10 copies per reaction, and this level of sensitivity is high and comparable to standard PCR assay. And when we are ready to perform ligation, we just flip the tube to release the salt that deactivated the polymerase, and at the same time, trigger the ligation. So we tested this with ELISA and were able to differentiate between 0% and 10% mutation. So that's good. So now the last part, in order to enable paper-based detection without opening the tube, we had to modify our lateral flow device to have sample ports with a plastic thumbtack at the bottom. So the tube that contain LDR product and the detection reagent can be loaded on and pushed to release the fluid onto lateral flow. So based on this, we are able to actually maintain, ambiguity within the cassette. So what I just showed you is the summary of the that I just showed you the 3 MVP total. We developed our first 2 MVP, OLA simple for DNA and RNA, and transition to the large scale manufacturer to generate sufficient prototype for our studies. We first validated MVP1 DNA on cross-subtype R-type specimen and showed nearly perfect performance. We also demonstrated the comparison with the result for RNA with the WHO certified lab result and showed 97% success. We also conducted usability testing both in the US and Kenya, both show 97% success. One thing that I forgot to mention is that the Ebola, actually we have 4 different versions, and I just told you one version of it just for the sake of time. And maybe one question that you have in your mind is that you have this picture of fully integrated device where we are on that. So it turns out that we are able to achieve several technology milestones. We were able to conduct on-paper molecular reaction, multiplex detection of SNP on a single device already, and also have a nearly automated paper network that can allow for something like this to happen. So you can imagine you have a network of paper and have lyophilized LDR on paper. After LDR product is done, it's loaded onto the device that allow for LDR to occur on paper at 37 degrees Celsius, then release the detected gold solution that allow for visualization of the multiple SNP in a single device. So I was quite proud at the end of my PhD that our work got highlighted at different places and also listed as the technology pipeline as part of the World Health Organization. And importantly, when we published our first work that include development, validation, and usability in one paper by Lancet, it published along with the editor choice of papers stating that our work actually made significant impact for HIV resistant testing field. And that was around the time in 2018. I was getting ready to graduate, and as you know, COVID hit. So it's an option of me to graduate right away or join the army to fight with COVID-19. So it turns out I selected the latter because it's the chance for me to really test what I have been growing my skill to fight with the pandemic, right? So it turns out the technology that I work on have been adapted to many other applications beyond the COVID-19. I won't be able to go into the detail, but just want to highlight a few things. So first step that come to mind is to develop lyophilized reagent for PCR for COVID. And we were able to not just use it on UW campus, but expand it across the country to Duke University, Georgetown University, and also across the globe to Zimbabwe University. Additionally, we also developed a point of care Harmony COVID-19 test in the Lutz lab. And the technology itself was licensed and now being commercialized as part of startup. So we received the FDA EUA approval, for example. And I also apply my knowledge for lateral flow strip to help the other field lab to develop their antigen-based test for COVID-19, and also offer my molecular knowledge for general molecular assay to help detecting COVID-19 in Seattle Metro bus. And the whole OLA Simple platform was adapted for detection of SARS-CoV-2 typing and also human drug sensitivity, like the example I showed you, and also use of both the context of healthcare in terms of genotyping cod fish for conservation purposes. So I have few last slides just to highlight. This is a lot already, but most of the work was done during my PhD. And my goal is to pursue a career as a faculty myself. So over the past year as postdoc, I didn't do much except writing grants. So I wrote 16 grants and I got awarded six proposals total. So I started to build my own research program that focus on 3 things that I care about: innovation, inclusion, and sustainability. And I didn't have the lab, but I was fortunate to be partnering with a researcher. So right now I'm based in Global Health, but I work with 8 different departments that have the lab space for me. And I have students from different departments to work on different parts of the project. So these are some of the pictures of me and my students. And I'm going to highlight maybe just a teaser because some of these projects are very early, but I just want to show you some direction. And maybe a year from now, if you have some empty slot for seminar, I would be really excited to share with you some updates. So here are the 4 domains of my research program. First is to an add-on of what I have done before for OLA Simple, for example. Coming from Thailand, I look back at a lot of technology, how it's developed mostly in the US, being evaluated in Kenya or Thailand. Then the developer in the US come back and make a lot of money. And sometimes the test ended up being so expensive that those people who were involved in the evaluation not even benefit from it. And sometimes it's not the fault of us, but the government corruption actually increased the cost by 3 times, for example, for the case of Thailand or transportation. So one domain that I try to do is try to develop a new technology transfer approach that engages local people to not just be the user, but they can be the co-inventor with us. So now I have a few projects that focus on supply chain development. So they learn how to make their own enzyme locally. They expand their manufacturing capability and try to write the training grant for the student locally. So then they actually can contribute into technology development. And then for domain 2, this is using the isothermal expand the usage of the test, not just for human, because we are living in a planet that have to focus on the health of the planet, that animal and also environmental in general. So this is the work that I collaborate with Homeland Security. It turns out that from the cod, there is a lot of illegal fishing that got caught and shipped to some other country and we imported back to the US. And cod is just an example, but there are so many other valuable wildlife that got done that way, and it's involved a lot of networking. So my technology that used for healthcare is now repurposed for law enforcement, and also that's how I was starting to collaborate with forest biology and also for zoonotic diseases in Thailand. For domain 3, this is focusing on new engagements. So I'm not sure whether you heard about this technology before, that you can use body temperature to carry out the reaction that perform with the quality just like PCR. So I wrote a few grants that focus on variable device to detect HIV viral load and also cytomegalovirus and also other STIs. But the focus is not on technology development per se, but really engage with clinician and patient to get their feedback of what is the form factor of the test that need to be designed in order to be adaptable and usable, adaptable by the local. And the last one, this is super new. I launched into a totally different area where I start to use synthetic biology and phage to try to trick bacteria into doing cell diagnostics. So a lot of work are still in the early stage, but it's very exciting because if it's successful, it could mean you can just add the sample into the tube that mixed with this chemistry, incubate for 1 hour, and the bacteria just spit the reagent that allow us to detect host, like more than 10 plus bacteria at once. So that could be something that really changed the field. So with that, I usually have a lot to thank. If you have time, you should check out on my website. I have 22 pages of my acknowledgement for my thesis. But here's just a few names that have been critical to my training at UW, and also my collaborator outside UW. funding agents that have been so supportive that allow me to explore different venues of my research, including discovery fellow that allow me to explore the work with Stephen Quaid and Shiri at Stanford during my PhD. And on the right here, these are funding that support my work. And one thing that I really glad that UW have this program is the scholarship for undergrad researchers. So one thing that keep the undergrad students excited about the work is that when they joined the lab first, they said, what made you excited about the work? I tried to design a project tailored for their need. And then within 3 months, they will develop their own abstract and essay that they can start, applying for their own scholarship. And that's actually, it's a way that you keep feeding on growing the next generation without just telling them what to do, but make them think about what they need to do. Well, I can talk about this all day long, but I think I should wrap up because we have only 8 minutes left before 1 p.m. But thank you so much for your attention, and I'm happy to take questions.
Thank you so much, Dr. Panpradist. I'm working on your name. I apologize. That was a great talk. I'm sorry I missed a part of it. We actually just had an earthquake here in Rwanda, so I had to stop and kind of pay attention to make sure everything was okay here. But 2 points from your presentation that I was really interested in. One is I really appreciate your perspective on having something to be locally developed and affordable, because that is inevitably the problem with any innovative device or equipment or software that we develop, especially if it's developed in the US. It goes down this pathway of getting FDA approval, which then makes it really expensive, and then you have to spend all this time recouping all of those costs. And I wanted to hear about how you thought, how you would address that, going forward, or what recommendations or suggestions you would have. The second thing is more sort of application related. A lot of our group is interested in surgical care and surgical care worldwide, and some of whom are probably thinking that I'm completely crazy and inviting you to speak in our group. But the challenge is that there are a lot of medical issues that surgeons have to worry about. For example, antimicrobial resistance is a major problem worldwide. And it seems to me this kind of technology could be very helpful in addressing what the resistance patterns are to then inform supply chain and be able to manage resources much more judiciously in places that don't have very many resources. So those are my 2 points, 2 questions, comments. I'd love to hear your thoughts. So thank you so much for joining us. I'm sorry I couldn't be there in person to meet you.
No, I'm really glad to be here. So maybe on the second question, one thing that actually I'm working on right now is trying to block cytomegalovirus detection, right? And I think there is a direct application for transplant. If I understand correctly, you have to screen for cytomegalovirus before you can do transplant. And currently it's not being screened. So you can imagine low to middle income country, that's a real struggle. So if they have access to antibiotics, prophylaxis, that's great. But if people already have the resistance in the background, that's also another problem, right? So I think just to close all the loop, one thing that I didn't mention is that, do you know that there are a lot of aquaculture that grow this seafood that we imported in the US that use a lot of illegal substances, including antibiotics? So there are the prawns that when we eat, in sushi or whatever, it's already loaded with antibiotics. So that's something that I try to work, as part of looking at a program, not just one is actually get into the human health, but look at it as a whole, One Health. Can you actually prevent those to actually get into our body first? And I think this makes it super exciting, because the thing is that wherever there is a DNA, there's a way. Or even right now, even though when you work with small molecule, you can always use the DNA to prick and use aptamer or whatever to detect those small molecule, and then use the back end process that I develop as a platform. So I always up to learn new programs and love working with different group of people. And now going back to the first question, sorry, I answered in the wrong order, but I think that's something that came to mind right away for the transplant. For the engaging with local community, I value this very much. And I think part of it is that one thing I saw is through HIV treatment. So HIV in the US is subtype B, right? Thailand, where I'm from, also majority subtype B. And there was the time that the US drug company rolled out the HIV treatment in Thailand. So they conducted clinical trial, they completed it, and then they brought back the result, got the FDA approved. But at the end, when it comes to implementation, it has to do with policy. So the way that the global structure is doing right now is that there's a country into low income country, high income country, and only the country that is low income country will be considered as part of the patent pool that didn't have to pay expensive costs for the drugs. So now you see the country like Thailand that's involved in the evaluation of the test, gone through the whole clinical trial, but at the end could not afford the drug itself, right? So that's one problem. So I think as a whole, what can we do to just start with educating people to realize their own power, that before you agree to accept the money to do clinical trial, what can you do to actually create equitable health medical research, right? So that's the first step. And to do this, it takes a lot of us. It's not just me. Many of us, like when we travel, the way that I got this collaboration, it just starting with listening, and I engage with them. So when I went to implement Ola Simple at Cambridge CDC in Kenya, I saw one person wearing really nice and red shirts, like, you have a cool t-shirt. And then we just started a conversation. And I told them that I'm here to implement Ola Simple for HIV drug resistant testing. Right away, the junior scientist said, we need one of that for TB. So I said, what do you want to do with it? So I just let them talk and hear their imagination. But I think what I can help is just, instead of defining what they need, we got to listen to them and hear what it is. So instead of pushing technology to them, we want to pull them to be able to develop technology. So then that was the beginning. On the weekend, we met and we talked about, okay, we're going to write them together. I'm going to list you as co-investigator. You are going to be co-inventor instead of the user. And you know how much it means to them. These are junior researchers. They went on to the higher up and convinced, please come talk to Nuttada. We don't have a lot of money. They didn't ask for like a lot of money, but we see the bigger picture of when they actually have sustainable tests that can be developed in their own country, it could mean a lot to them. So then they identify, the whole workflow. So instead of developing technology in the US, go to the whole regulation or whatever, they said, no, we're going to work with local. These are the key organizations that we need to engage them at the get-go. What are some things that we have to consider to make sure that in first prototype or minimal version of changes, we can actually get the product to local, being made by local, understood by local, distributed by local, and at the end, if we can like duplicate this effort, I think that's how we're going to solve the problem. Honestly, we won't be in COVID-19 pandemic right now if each country has their own capacity, and I think we need every single one of us to do this. Sorry, it's just really real question. I hope that answers some of your questions.
Yes, absolutely. And I love that philosophy of listening and being open and engaging and drawing out their, other people's needs and building something based on that, and that equitably involves them. I love that concept, and we hope that we also have that kind of a mentality in Utah. Anyway, I'll turn the conference back to Dr. Ng. I don't know if he has any questions or closing comments.
I don't have any questions. I'm very thoroughly impressed by all your accomplishments. I mean, just 15 months in a postdoc, and you're running it's like you're running a mini research group already. And when I was a postdoc, I didn't even know how to do that. Wow, that's amazing. Thank you so much for your time. Thank you so much.
Very nice to meet everyone. And the barcode here actually linked to my website. I use many types of social networks, so please stay connected. And I should mention that one of the projects that I was talking about enzyme production, I collaborate with one faculty in Utah already. So I think it's just about growing the network from here. And if you have ideas, I'm always down to collaborations because we share the common goal. So, yeah, thank you so much.
Okay, thank you, and good to see everybody else. Okay, take care. Bye-bye.
Well, thank you.
Thank you.